This method can help answer questions in the field of plant glycoscience, such as how N-glycans of plant origin can be quickly analyzed and characterized. The main advantage of this technique is that it doesn't require any buffer changes between the enzymatic release of N-glycans and the derivatization reactions with 2-aminobenzamide. Demonstrating the procedure will be Ms. Yamin Du and Ms. Shen-Li Zheng, two graduate students from the Glycomics and Glycan Bioengineering Research Center at Nanjing Agricultural University.
To begin isolating the proteins, use a kitchen blender to homogenize approximately 100 grams of fresh radish for 10 minutes. Transfer the resulting slurry to a 50 milliliter centrifuge tube. Centrifuge at 14,000 times gravity at 4 degrees Celsius for 20 minutes to remove the insoluble material. After this, carefully transfer the supernatant into a new 50 milliliter centrifuge tube. Add an equal amount of a 2 molar trichloroacetic acid solution. Centrifuge at 14,000 times gravity at 4 degrees Celsius for 30 minutes. And then, remove the supernatant. Wash the pellet with 20 milliliters of deionized water. Centrifuge again at 14,000 times gravity at 4 degrees Celsius for 5 minutes to remove soluble oligosaccharides and polysaccharides. Then, resuspend the pellet with 1 milliliter of deionized water and transfer the protein solution to a fresh 1.5 milliliter centrifuge tube.